Thank you. So I will be talking about polymorphism-ware phylogenetic models and their applications. So the idea when we developed these models really was that we were trying to combine different evolutionary processes, different levels of modeling in my lab. And one level is the phylogenetic level. So here you see a phylogenetic tree that visualizes the relationship between primates. And you're well aware that obviously we're not only having sequences from one human, but we have, at the time we started, there was already a completed 1,000 genome human project. But now there's projects like the 100,000 UK genomes and other populations. So really the idea was, can we link these evolutionary processes and model evolution at different time scales at the same time? So when we started, at that stage, there was already multiple ideas around how to combine multiple individuals for constructing evolutionary histories. And yesterday we had this excellent talk by Si-Heng Yang explaining the multi-species coalescent. So obviously my life is now easy and I can squeeze that in half a slide. By the time we started thinking about the problem, people were aware about incomplete lineage sorting and problems that could cause. So here I visualize another case of incomplete lineage sorting. And the multi-species coalescent was a solution for that. When we looked at it, we thought, yes, this is really well describing all possible genealogies and the phenomenon of incomplete lineage sorting, but you can't really do this for many populations. So it's accurately describing it, but it's rather slow. And there were more genomes coming out, again, multiple individuals per species. And the probably naive and rough approach is simply to take this genome-wide data, concatenate it, just take one individual per species and reconstruct a species tree. So this would be super fast and was possible by using a standard phylogenetic software like FireML, RaxML, and IQ-Tree. But it totally ignored the possibility of having these multiple individuals. And we were trying to come up with an approach which was intermediate, meaning that we wanted to account for multiple individuals, but we were not coming from the populations of the, and multi-species coalescent side, which is very computational intensive. Instead, we were trying to model effects of these different genealogies and the incomplete lineage sorting. So if you look at the multi-species coalescent and you have a mutation, for example, here in the tree, what that means is you're going to observe polymorphism at the internal nodes, as well as the tips. And you could account for this by taking into account all these possible genealogies, or you could just say like, well, we just model shifts in allele frequencies along the tree. And that's the basic idea of the polymorphism-ware phylogenetic models. There are phylogenetic models. So in this sense, what you need for a phylogenetic model is you want to construct a tree, and you also need a substitution model. And the substitution model is often a continuous time Markov process that is defined by its instantaneous rate matrix. So here you can see an instantaneous rate matrix. It's basically just listing the rates of replacement from between the different nucleotides. And once you have that, there's multiple software packages around which are very efficiently calculating likelihoods for trees. So what we we're planning to do is, instead of going directly from one nucleotide to another nucleotide, so having a substitution from C to A, we're going to break down this process and have a mutation and the population genetic processes of genetic drift and selection. So polymorphism-ware phylogenetic models break down the substitution process into the constituent population processes. We account with this for existing polymorphisms, but we have to work with a new state space. So this is an example here of such a state space. You will have fixed states, monomorphic states. So for example, here, all the individuals have an A, which is also here colored by red, so the red allele. And then you can have a mutation and a blue allele, and this blue allele can increase and decrease in frequencies, and you would have intermediate polymorphic states with different allele frequencies of the A and the C allele. We know how to move around between fixed states and how to introduce mutations, but we also need to model how these allele frequency shifts between the polymorphic states. So how do we do that? Well, the mutation, we can use a standard model that we know from phylogenetics already, like HKY or GTR. The mutation can only happen at the boundaries. What does that mean? It means you can't jump to high frequencies with one step. You always have to start in a fixed state and introduce the mutation. This is kind of a bit sloppy way to talk about this. And in the morning, there has to be a practical. So if you wanted to look at the slides, it's a really nice explanation. These models, because you can only come in from fixed sites, are sometimes called boundary mutation models. And there's elegant mathematical ways to describe that. And actually, Dominic Schrempf and Asker Holbrook have a really nice paper on boundary mutation models and their advantages. So once you have the mutation, we would like to move between the polymorphic states, and we do this by modeling genetic drift with a Moran model, a classical population genetic model. It's a birth-death process. So let's do an example. So here we have the blue allele, just introduced by a mutation. We could increase the frequency of this blue allele by picking an individual with a red allele and let it die. The probability of letting this individual die, it's random, we randomly pick it, and the probability for that is n-1 over n. And to give birth to the blue allele, the rate would be 1. The process is symmetric, so we could also lose this blue allele. How would that happen? Well, we would pick with the probability 1 over n the blue allele, let it die, and with rate n-1, we would give to a new birth to a new red allele, and we would be in the fixed state again. So the really nice thing here is this is symmetric, right? And it allows us to move between these polymorphic states. So having now defined how to introduce mutations and how to move between the polymorphic states, we can write down an instantaneous rate matrix. There is no big shifts in allele frequencies, so all the rates which would account for bigger shifts are 0. At the boundaries, with a mutation model of your choice, you can introduce a mutation. And between the polymorphic states, once the mutation is introduced, we can move with a Moran model that is symmetric. The good news really is once you have an instantaneous rate matrix, you can do phylogeny. And we increased the state space. So now, obviously, this comes with a computational cost to have a larger state base, but it's actually not so bad. So to make an example here, this would be a virtual population of 10 individuals. We have 4 fixed states for the nucleotides. We have 6 possible allele pairs, and intermediate state counts, we have 9. So the total size of that state space is the 4 fixed states and 6 times 9 polymorphic states. That's 58 states. That's about the size of a codon model. So if you can use codon models to infer phylogenies, you can use polymorphism-ware phylogenetic models. So really, so obviously you can play around with the discretization here to mimic the dynamics and the number of polymorphic states. But in principle, this was a way to model polymorphism, manageable in standard phylogenetic software packages. So the advantages of POMO are really that we now account for the ancestral and the present polymorphisms. There's many nice mathematical properties. The model is reversible. We can write down an explicit stationary distribution, and it can be implemented in a maximum likelihood in a Bayesian framework. We had this a couple of times already in the workshop. So once you have a probabilistic model of sequence evolution, you can use them in both frameworks, because for both frameworks you actually would need the likelihood. I'm going to start off with the maximum likelihood implementation of POMO. So Dominic Schrempf, who gave the workshop in the morning, he implemented the POMO models into IQ3. We've chosen IQ3 for several reasons. One is, well, this is also a virtual, but it's still a Vienna workshop. And at that stage, Dominic and Min Bui in Arndt van Hessler's group were both working on IQ3, so they could exchange each other. IQ3 also has many nice features. There's various substitution models that, and you get help to choosing them by having a model selection. There's rate heterogeneity, bootstrapping, and it's fully polarized software and many other features. From our perspective, the most important feature is, of course, that it has polymorphism-aware phylogenetic models automatically now, actually, if you download a version that is higher than 1.6. So most of these rather more complex models that you can combine with polymorphism-aware models, that would happen automatically. But sometimes you have to think a little bit more in detail. So one thing which is very popular in phylogenetics is to account for rate heterogeneity across sites. POMO models can do this, but you have to think a little bit harder and break down the instantaneous rate matrix of the POMO model into a mutational process and the drift process, because the rate heterogeneity would affect the mutation rates. It shouldn't affect any of the dynamics of the drift, right? But you can split the matrix into the mutation part and the drift part, and then only apply the rate heterogeneity to the mutational part. So that's one of the examples that POMO can be used with all kind of different models, but sometimes you have to think a little bit more careful how you adapt the POMO model. We wanted to check if it's any good, so we did simulations. Usually, if we test POMO, we try to use a model that is not too similar to the POMO model itself. So we used SymPhy, and we created, in this example, I'm going to show you really a large tree with a Yule model and a birth rate such that we would expect about 100 species, which 10 individuals each. So if you think of the multi-species coalescent, if you wanted to do this, this would be a tree with 1,000 tips, so that's quite big. Once we had the trees, we used them to generate sequences and using section. So I said we kind of try to use SimPhy, or we also used other coalescent-based simulators. So I would like to encourage you, we also have our own simulator for POMO, to use that actually if you do coalescent simulations, if you wanted to misspecify and you don't want to do forward simulations. It's well worth having a look at this simulation model. It's called BMM for boundary mutation models, and it's with an IQ tree. So this is CPU time. We first, before we did IQ tree, had a version in Hi-Fi, which was already faster than some of the multi-species coalescent models I told you in the beginning, like B-star and BEST. But IQ tree really helped to make the versions faster. So what I'm comparing here in gray and in black is the increase in CPU time on the y-axis if you take more genes. Gray would be just taking one individual, an HKY model, and for all the genes you use concatenation. For IQ tree, you also use the concatenated alignment, but it's multiple individuals and it's summarized. And we used ref POMO. So what you can see is taking more genes. In fact, actually, obviously POMO is a little bit slower, but at the end, it almost looks like POMO and concatenation have the same speed. So I was a bit surprised about this because I told you at the beginning you have to increase the state space. So it should be always a bit, can be fast, but should not be as fast as concatenation. So I think what's happening here is because these trees are simulated on the assumption that there's incomplete lineage sorting, that's actually rather hard for IQ tree to converge. While RefPOMO doesn't have that severe problems with it, and that's the reason why it's almost as fast as concatenation. So basically what I'm trying to say with this figure is, before you do concatenation and you know there's a lot of incomplete lineage sorting in your tree and you have lots of species, you should really rather use a POMO model because it's not going to take you much longer. Is it better? So the problem with incomplete lineage sorting and concatenation has been shown that the error doesn't go down when you increase the amount of data. So here we increase the data with the number of genes. The typical gene length would be 1,000 nucleotides. The error is measured by branch score distance. So that measures differences in topology, as well as differences in branch length. So what you would like to see from a method is, like here POMO, if you increase the amount of data, hopefully your branch score distance, the distance between the true simulated tree and the one you're inferring, should go down to 0, or at least decrease. For concatenation, this is not the case. And this is known to be problematic, especially in cases of incomplete lineage sorting. So we can do better than concatenation. We are almost as fast. Other parameters, so branch score distance, I already showed you, BSD. In this example, we also inferred kappa, the transition transversion rate ratio in the HKY model. The level of heterozygosity, theta, is also well measured. So this is the just test value. It's the error, the relative error here. And alpha, that would be the alpha of the rate heterogeneity, the gamma distribution. That one is not as good, but you can see the relative error is not going much over 5%. So it's actually still a pretty good estimate for this parameter as well in the simulations. Time for real data. So I'll show you actually, we've tried that with a couple of standard data sets which are currently around, but I actually thought I show you one which is typical, but it's a rather hard case. So we were involved in the Baboon Genome Consortium. The good thing with consortia is that several groups work on the same data set with different methods. So we had additional information to what we could do with this data. So the baboons are interesting because they have still ongoing, differently to humans, still ongoing hybridization zones between these different populations. There's many of them, but the little X's on this map of Africa show the ones where they're documented in the literature. There's two clades in this baboon data, the southern and the northern clade. And in Aarhus, the group around Thomas Myland, and I also mention others which were involved in this analysis, they inferred that the Kinde species is the result of an admixture of the northern and the southern clade, probably almost 50% here. So given that little of a mixture, we thought like, well, this is not something we explicitly model, but maybe we try POMO and see what's going to happen. So we used whole genome data, extracted the fourfold degenerate sites from the orthologous alignments. It's not as many individuals as what I told you we can do in the simulations. But it's multiple individuals per species, and because it's not so many populations, we were able to do bootstrapping. And this is the result. So in some ways, it's nice. It nicely splits off the southern and the northern clade. If you would do gene trees and try to summarize them, you could do this, for example, by just beginning like initially with something called concordance factors. So how many times, if you take the individuals from this data set, how many times would that match the tree which is shown here? So the problem with the concordance factors is that you actually then would only find for the gene trees about 14% would match this clade. But if you take the alternative clade, it's actually even worse. And the same for the southern clade. So you actually don't find many trees which agree if you just take the individuals gene trees. While if you use POMO, you get a relatively clear picture. Almost, if you look at the bootstrap values, almost, to say we're a little bit overconfident, but it's also because there's so much data. So we're quite pleased by this result. But we also at the same time still were a little bit puzzled. So what puzzled me was that these species are recently diverged. They probably still have around the same generation time. The selection pressures are probably not so different. So why is there this, for example, this Ursinus branch, which is so much longer? Why is there accelerated? It looked to me like accelerated evolution. And I wasn't really sure if I would understand this. So we did some simulations because we had information about the admixture. So we increased the level of admixture between the north and the southern clade going into Kinde. And if you do this, the total branch length of the tree estimated in POMO is not changing. And when we looked at the tree, I first thought like, the Ursinus branch actually increases, but root to Ursinus doesn't increase either. What does change is the root to Kinde branch. That one is getting shorter, and in some ways, it's something you have to consider if you do this analysis and you think about gene flow. But it kind of makes sense because if you think of branch length as genetic distance, and you now have this strong admixture coming from the other clade, it probably makes sense that this branch is getting shorter. It's obviously also reflecting that you have all this shared polymorphism because of the admixture between these the Skindey branch and the northern clade. Maximum likelihood part of the talk. I just to encourage you, it's user friendly and do try it out. It makes sense. We would recommend it with more than 4 species. If you have less and other questions, you might actually consider obviously using other softwares like multi-species coalescent processes. But in principle, we could easily run it for 100 species and more. Another reason, actually the original reason why we were choosing to have polymorphism-aware models is, and this is now really something which is very difficult with multi-species coalescent processes, is to consider selection. And it almost happens naturally if you do POMO models, because there's the Moran model involved, and the Moran model with selection is a process which is already established and also rather well understood, actually. So how does that work? Let's consider selection too. So we introduce a mutation. Now, once we have the mutation, well, can we increase the blue allele further? So to increase the blue allele, you have to let a red an individual with a red allele die. So that would again happen just randomly. So the probability for that is n-1 over n to let the red allele die. Now, if we consider selection, to increase this blue allele, the rate is not only 1 plus, but 1 plus the selection coefficient, the fitness advantage of that blue allele, which we would denote with the parameter sigma AI. So basically, you just have to introduce this allelic selection coefficients, and you have a model which is polymorphism aware, which includes selection. So it doesn't, obviously the instantaneous rate matrix, when we write it down, it looks a little bit more complex, but it's actually not so much more complex. We just have the only additional parameters which we have are the selection coefficients. So you still have mutation models to introduce your new alleles. You can't have larger shifts, so all the larger shifts have a rate 0, and then you just have the selection coefficients for the different alleles. So no reason not to include that in a model. And we wanted to do Bayesian models as well, because we wanted to understand the effect of including selection on molecular dating. And Bayesian models are far more suitable for molecular dating. Probably heard that during the workshop several times too. They're slower. So while we were rather kind of, not too focused on how many intermediate polymorphic states we should have originally, we now, given the harder computational tasks, thought like, well, we should really think how many do we need. The good news is you can mimic the dynamics already with a POMO2 and a POMO3 model. So the way we do this is we know the level of variation in our data, the heterozygosity, and we fix that. And then this allows us to scale between different virtual population sizes. And the relationships are rather easy. So for POMO2, this is a model which allows you to include drift already. So we, in this visualization here, in the vertices, we have the fixed states, and on the edges we have polymorphic intermediate states. So with this one, you already have a polymorphic model. It only has, it's only a POMO2 model, but it allows you to include drift. With two states, you can do this. The relationship between larger population sizes than this virtual POMO2 would be just using the harmonic number here. So you can actually write down the relationship between the mutation rates of the virtual population and the effective population, larger population. For having selection, you need a POMO3 actually. So you have to have one more intermediate polymorphic state to model this. The relationships between the mutation rates, between these virtual populations and the larger populations get more complex, but we can still write them down for the mutation rates as well as for the selection coefficients. So basically what I'm saying is with the POMO2 and 3, you can already mimic the dynamics of having drift, and for POMO3 of having drift and selection. Does it make a difference? So we first did simulations.
Carolin, I'm just going to let you know that there are many interesting questions in the chat, but if you want some of them, then you need to finish off.
Okay.
Well, there are 5 minutes left.
There are 5 minutes left? Okay. So I summarize this. It does. So does it make a difference? In our simulations, what we are showing, we have simulated data sets which are similar to flight data sets and grade 8 data sets. Basically what you see, this is a Bayesian method, and the question is how often does the map tree matches the true topology. If you increase the data, actually, if you include selection, it does help for less sites, but basically, if you have enough data, you're probably going to get the right topology. Where it gets really interesting is if you look at the branch sites, at the branch length and the error there, because what happens there is with standard GTR models and the model which allows for polymorphic states and which allows for selection, you really see a difference, and it does make a difference. So to summarize, this is real data. It's Drosophila species. And the Drosophila species are, it's 31 populations. And we inferred the trees. What you can see between the POMO3, which allows for election, and POMO2, which is just neutral evolution with drift, is that the branch lengths are slightly longer. So, for example, if you look at the purple clades, so what that is, the split there, it's the split between the populations which are out of Africa and in Africa. If you have selection, this is something which is over 30,000 years. If you don't have selection, it's slightly under 30,000 years. Actually, an age under 30,000 years is also what is found in the literature. So it does make a difference, not so much in topology, but clearly, if you estimate a branch length to include selection, and this could be very weak selection, so it doesn't, it's just because something like biased gene conversion, which is weak, but is very persuasive because it affects a lot of sites. So this is what is reflected here. So I'm not going to say much about this. I thought, there's another level, but, people can come to us and talk to us. You could also use these polymorphism-aware models for experimental evolution, where you could estimate the selection coefficients using the Moran models of time series data. So that's another level in which you could use these kind of models. So and the advantage is really that you explicitly can calculate selection coefficients. You're not only finding targets of selection, you also can give the estimates for the selection coefficient. So to summarize, POMO accounts for ancestral and recent polymorphism. Selection can be taken into account. There's efficient implementations also for species trees, but also for experimental evolution studies. If you have more ideas about datasets, come and talk to us. And I'm obviously also very happy to take questions from the chat now.